Hello, everyone. My name is Mireia. I'm a third-year PhD student working under the supervision of Professor Anna Grabowska and Professor David Bates at the University of Nottingham, the Biodiscovery Institute. I'd like to start by thanking the AMMF for selecting my abstract and giving me the opportunity to talk and show a piece of my work. So I've always been really interested in cholangiocarcinoma, and I decided to pursue this as my PhD, ultimately with the aim to provide some new insights to patients and families that have been affected by this devastating cancer. And the focus of my PhD is the tumor microenvironment, and I'm trying to understand the role of mesenchymal cells in cholangiocarcinoma. We all know that CCA is an aggressive and heterogeneous malignancy. Treatments are limited and often ineffective due to the lack of understanding of its molecular pathogenesis. And it's also known as a highly desmoplastic tumor, but we yet don't understand the functional role of this reactive tumor microenvironment. Therefore, we believe that investigating the tumor microenvironment and its role in CCA progression could lead to more effective therapeutic treatments for managing this devastating cancer. 2D cell lines have been an effective preclinical model to study cancer. However, recently, more in vitro models, such as 3D models, have been developed to improve this 2D cell lines culture. 3D models can recapitulate features of the native tumor, such as the presence of tumor cell-tumor microenvironment and extracellular matrix interactions and gradient distributions of chemical and biological factors. Thus, they could be more promising models to study cholangiocarcinoma. We have managed to develop 3D spheroid models from either cancer cell lines and patient-derived xenografts, using ultra-low attachment 96 well plates. PDX are models of cancer where the tissue from the patient have been implanted into a immunodeficient mouse. So they have always been growing in a physiological environment, and they are known to retain features of the original tumor. We use these cells to form spheroids to further understand the pathogenesis of the cancer. We have characterized these models by live death staining and HNT staining. And we've also determined the effect of mesenchymal stem cells in driving proliferation by metabolic assay and transcriptomic analysis. We've done quite a lot of work, but today I will be only showing some of the main findings. Start by showing that CCA-derived spheroids stay viable and retain histological features of the CCA tumor. I successfully generated a big panel of cell line-derived spheroids. However, I'm only showing here 3 of the cell lines. So KKUM1, 2, and 3, KKUM156, and RB. Morphology differences between spheroid form were already observed at day 3 of culture and was more pronounced after 7 days. High compaction of cells was observed in all spheroids with a well-defined border. RBE spheroids, although forming a compact spheroid, the size remained constant over the time of culture. And it is also really interesting to see the distinguished behavior of the cells when forming a 3D structure. In addition, at day 7, cells remain viable, as we can see by the live death staining. So this was performed by adding a dye mixture to the spheroids. Calcium was used to measure metabolically active and viable cells as green staining, and ethidium homodimer 1 was used to indicate cells, dead cells, which is the red staining. For more in-depth analysis, HNT was performed. CCA cells were grown as spheroids again, and at day 5 they were fixed, paraffin embedded, sectioned, and stained with HNT. HNT staining also confirmed that spheroids display classical neoplastic features that can be observed in the CCA tumor histology. We also found that cell proliferation was found to be promoted by the presence of mesenchymal stem cells. HNT of the PDX tissues were with the presence and absence of mesenchymal stem cells was performed and compared to its matched tumor tissue. We can observe that the PDX retained the phenotypic features of the original tumor tissue. And this result, it is more noticeable in the presence of stroma, as we can see. But overall, we can confirm that the PDX model seems to be a reliable system mimicking the original tumor. Additionally, we generated a 3D spheroid in vitro model from this PDX, and a metabolic assay, Presto Blue, was used to provide a quantitative measure of the cell proliferation, and we can observe that the proliferation was enhanced by the presence of mesenchymal stem cells. This is a result that we've seen in the panel of the cell line-derived spheroids. And in addition, what we can see is that in the mesenchymal stem cells appear to help spheroid formation. So therefore, we can conclude that the mesenchymal stem cells which are present in the stroma are supporting somehow in vitro CCA formation and growth. Finally, we've managed to do an initial transcriptomic analysis to compare the PDX tissue with and without mesenchymal stem cells. Nonetheless, we're planning to do a bigger analysis. But briefly, I'll go through this result. So we perform a gene seed enrichment bioinformatic analysis from the RNA-seq from PDX tissues with and without mesenchymal stem cells. And we saw that CCA-relevant pathways were activated by supporting mesenchymal stem cells. Here we've illustrated genes that are involved in biological processes activated by supporting MSCs by a Venn diagram. And some of the relevant pathways were dephosphorylation, protein dephosphorylation, negative regulation of mitotic cell cycle, and negative regulation of EGFR signaling pathways. However, as I said, further analysis will be conducted with a bigger set of samples. So we can conclude that based on initial findings, 3D spheroid models appear promising due to their histological resemblance to the patient tumors. And in addition, this study could provide new insights in the crosstalk between the tumor and the stroma, ultimately providing promising targets for the treatment of this cancer. So thank you for listening.